Welcome to Interface. Today we have an eminent scientist, Dr. T.J. Pantin, who contributed immensely to the fish genetics and is the person behind producing the first Indian transgenic fish. Sir, we are familiar with the cryopreservation of sperms in humans and on livestock, but in your experiments you have shown that you can preserve the and revitalize the sperms from a cryopreserved motylized fish. So, could you explain how you did this?
Professor Kannan, cryopreservation is a very sophisticated technology. It requires a very costly equipment of about 30 lakhs.
Yeah.
So, in a big country of our size, we can't afford for that kind of a costly equipment, that kind of a sophisticated technology.
Yeah.
In Madurai Kamaraj University, we first time discovered cadaveric sperm. Sperm taken from dead specimen can also fertilize the eggs in fish. This is an accidental discovery. I was very particular to have a particular catfish with a particular genetic character. Unfortunately, without my knowledge, the fish died. Afraid of me, my student preserved it -20. After 180 days, I asked my student to bring that fish. He could not bring it. He said it is in deep freeze at -20 degrees. As you are aware, at -20 degrees, it is called biochemical zero, where no biological activity is possible. So, I just somehow wanted to produce a progeny out of that particular catfish with a particular characteristics. So, left with no alternative, we opened the animal, and to our horror, we found under microscope the sperms were alive and motile after 188 days. I also like to bring to your kind attention some general knowledge. Mammalian body has 257 different types of cells. We declare somebody is dead when the heart is stopped. But we don't know the rest of the tissues have either died or not. We are not aware. It takes long time for many other tissues to die. And perhaps the contents of testes takes very long time to die. And therefore, even after the death of the specimen, when it was kept in -20 degrees, the sperm was alive for 188 days. So, we use the sperm to fertilize a fresh egg, and to our surprise, the egg was fertilizable, young ones came. But the percentage of young ones which hatched out were less than 3%. We thought this kind of simple technology, because -20 degree, a deep freezer cost only 20,000 rupees. Freshly dead animals to be kept in deep freezer is a very simple technology. It can be practiced anywhere from Shillong down to Trivandrum. It can happen either from Delhi to Kanyakumari. So, we thought we will bring in a very simple design of preserving this fish sperm. Following our discovery, many other laboratories world over have confirmed that what little we have found out is correct.
Sir, again you have another great discovery for the humankind. I guess because of the human interference in the ecosystem, many animals and plants are getting extinct, and they move to the verge of extinction, some of the animals. And I understand that you developed a technique where you can make use of the sperm of a particular species, say an endangered species, and fertilize egg of a related alien species by inactivating the nucleus of the egg. So this technique will be immensely useful to propagate the endangered species. So could you explain, sir, how this technique works?
It's all known that a fertilized egg is omnipotent.
Okay.
Out of the fertilized egg, an individual can come out.
Okay.
But as the development proceeds, what was once omnipotent becomes pluripotent.
Oh, yeah.
And as still development proceeds, pluripotent becomes unipotent.
Oh, yeah.
So, in an individual, we have only unipotent cells.
Okay.
For instance, if you take mammals,
Yeah.
the mammary gland is unipotent because it can produce milk and nothing but milk.
Oh, yeah.
So, for the first time, Wilmot and his colleagues
Oh, yeah.
at Edinburgh
Oh,
cloned the so-called dolly
oh, yeah.
out of a cell from
The adult cells. Yeah.
adults highly differentiated cells
Yeah.
taken back to enucleated egg of a sheep.
Oh, yeah.
We could not do this in fish for several reasons, because the differences between fish and mammals are so different.
Okay.
So, instead of taking a highly differentiated cell, I took the sperm as a craft for introducing the genome.
Oh, okay.
We also, throughout our career, we wanted to use very simple, widely practicable technique.
Oh, yeah.
That's why we have a fish which is likely to become endangered
Okay.
or extinct
Yeah.
if we have the sperm preserved at -20 degrees.
Yeah.
We only need this sperm because it has a genome for that particular species.
Oh, yeah.
Now we can have egg from a related species. It could be within that genus, or it could be from the same family.
Okay.
We have not gone beyond the family.
Oh, okay.
So within that, if you take an egg, you can use UV irradiation technique. UV irradiation bulbs are very cheap, 20 rupees.
Yeah, yeah.
So you can practice it. If you expose the egg to UV irradiation for a few minutes
Okay.
or few seconds,
Oh, yeah.
the genome in the egg is silenced,
Oh, yeah. Inactivated.
Yeah.
Yeah.
inactivated.
Yeah.
In the case of Wilmot, it was eliminated.
Oh, yeah.
We are only inactivated.
Oh, yeah.
In the genome inactivated egg, if we introduce a sperm of the species which we want to recover,
Oh, yeah.
it goes on very well. Heterologous fertilization is possible.
Oh, yeah.
Young ones hatch out. That is using a surrogate egg of an alien species.
That's the beauty of it.
We can use the preserved sperm and restore the genome of the species which we want.
Oh, yeah.
This technique is not exactly the same by which Dolly was cloned.
Oh, okay.
It is little different. But it has some other use. Apart from its simplicity and wide practicability,
Oh, yeah.
it has other uses. For instance, in India, we have carps.
Oh, yeah.
Carps are annual spawners. They spawn only during monsoon period.
Yeah.
But if we want to have carp seedlings throughout the year, we can use tilapia, which breeds throughout the year.
Oh, okay.
And tilapia is also not wanted. And so, we can preserve the sperm of carps
Okay.
and use the eggs of tilapia,
Oh.
genome inactivated tilapia eggs, and introduce the sperm of carp and thereby produce throughout the year
Oh,
carps.
Oh.
This kind of several advantages. Not only that we are able to recover, restore the genome of endangered species, but also we can use this technique for commercial purpose also.
Yeah, right. Okay.
This is what we developed in Madurai Kamaraj University, and right now many other countries are following our techniques to produce, to recover the genome of fishes belonging to their country.
Sir, the transgenic organisms, genetically modified organism, is a hot topic nowadays for both for science and also for the common part. It's being discussed widely. For the scientist, it's a way to improve the crop yield so that we can address the question of hunger, and also scientists are looking to improve the method. But for the common man, there are several concerns also. So, being the first person to produce a transgenic fish in India, before going to those aspects, could you just explain how you produce this first transgenic fish?
I like to bring to your kind attention. Once you middle with the genome, it becomes a transgenic. Middling can be of different degrees.
Okay. Yeah.
What we have done is, Dr. Kannan, as you know, the growth, somatic growth of our body depends upon a hormone called growth hormone.
Right.
The growth hormone is controlled by a gene called growth hormone gene.
Okay.
So what we have done is, we have not taken any foreign gene of another fish or another animal
Okay.
to introduce this into Rohu. Second thing, that we only increase the copy number.
Okay.
We have not disrupted or changed the genome of any sort.
Oh, okay.
Let's say the Rohu has 10 growth hormone gene copies.
Okay.
We introduce another 10
Oh, okay.
to make it 20 and to see whether the fish would grow 2 times faster.
Oh, okay. Yeah.
This is the kind of transgenic fish that at Madurai Kamaraj University was produced for the first time.
Not a foreign gene.
Yeah.
Yeah.
But also, this growth hormone gene, to prepare that transcript, you cannot go to a different country and bring them.
Okay.
It cannot be imported.
Okay.
We produce for the first time a Sudesi, indigenous transcript
Oh, okay.
consisting of our own Rohu growth hormone gene.
Okay.
That took long time for us to clone,
Okay.
sequence, and then put them in, you need a promoter, this and that, so many things, so that the introduced piece of transcript can be attached to the organism's genome,
Okay.
and it can express.
Oh, yeah. Yeah.
I like to also bring to your kind notice, there is a vast difference between transgenic plant and transgenic animal.
Okay. Yeah.
In transgenic plants, you don't have a brain,
Okay.
nervous system. So whatever happens to the gene, it is immediately expressed.
Okay.
For instance, if you have a seed,
Okay.
if you produce triploid,
Okay.
the plant grows 3 times faster. If you introduce a triploid fish, it is not growing 3 times faster.
Oh, okay.
Because there is something called nervous system, and the nervous system tends to control either the total number of cells
Okay.
or the size of cells.
Okay.
So even after introducing these 10 copy numbers of growth hormone gene in Rohu, the growth was not doubled, but it increased only to 1.6 times faster.
Okay. Yeah.
But still, it did it.
Okay.
But in a country of our size, with different religions and different beliefs, etc., transgenics are not yet accepted.
Okay. Professor Pantian, so people have several concerns about the transgenic animals and plants. And one of the concerns is regarding the presence of this transgene or the foreign gene expressed in all parts of the plant or animal. Say, for example, in a particular fruit plant, if you eat the fruit, it's not the fruit alone, but all the plant parts are expressing the same gene. For example, Bt toxin. and it may pose a health hazard. So it's a concern, and one way I understand that scientists are trying to express it in wherever it is necessary. So how far we are now succeeded in limiting the expression of this transgenic gene to the required part of the plant or animal?
Professor Kannan, I think there is a general wrong impression by common man as well as scientists. Time and again we have hybridized many plants, many animals. Once you hybridize two different species, either in animals or plants, we have already dampened with the genome.
Okay.
And the hybrid products are being very common, very easily, readily accepted by common man as well as by scientists. When it comes to transgenic, people have apprehension. I like to bring to your kind notice, 10-12 years before, Government of India, with the concern of scientists and our own parliamentarians, introduced transgenic cotton.
Okay.
The reason is, we are not going to eat the transgenic cotton. We are only wear the cloth.
Yeah, right.
But you must come to know that the cotton seed is being eaten by our own cattle.
Oh, okay.
And we are drawing milk from them.
Okay.
Cotton seed oil is exported to Europe from India. Europe is a country which has lot of apprehension for transgenics.
Yeah, right, right. Yeah.
Even so, common man has accepted transgenic cotton for the last 10 years. But when it came to transgenic brinjal, people had lot of apprehension. Our own ministers also had lot of apprehension. This is simply for the reason, unlike cotton, brinjal is taken directly by us. Therefore, I readily agree the common man's concern, minister's concern, that how can we accept. But I think once you understand that we don't hesitate to eat a mango, which is a hybrid product, why do we have any apprehension for a transgenic brinjal? A country of this size, we need huge quantum of food grains, pulses, vegetables, and so on and so forth. The land mass available for cultivation is limited and constant. But the population, if you would permit me to say, in 1947 we were only 20 million. Today we have to feed 120 million people. That means from the available land, we have, our productivity has to be increased. And therefore, we have to hybridize, we have to bring in new strains, etc. Take for instance, these short varieties, dwarf varieties, is also genome has been manipulated, which we have accepted. We have been once producing 50 million ton. Today we are producing 260 million ton to feed 120 millions of Indians. I think the primary job of a government, primary job of scientists who are supported by the government money should be to see that every Indian has enough to eat. Eat healthy food. Once it is proved that a transgenic brinjal is harmless, I think we should have no hesitation to accept it. We should not go by superstitious feelings or religious or all kinds of feelings. Once scientists clear, I think it should be accepted by common man.
Professor Pantin, another great concern for the ecologists as well as for the common man and the farmers is that the transgenic crops may contaminate the wild varieties and natural varieties of crops. Say for example, if you cultivate the BT brinjal, the pollen from this BT brinjal may go and fell into a normal brinjal, thereby contaminating the genome of the natural variety, and in that case we will lose all our natural varieties of genome. So what is your opinion on this? How we can do or prevent this, do away with it?
Professor Kannan, I really appreciate your concern for a common man. This can happen, and this will happen.
Okay.
But please understand, Government of India, under the system of Indian Council of Agricultural Research, have established a Plant Genome Conservation Center at New Delhi, Animal Genome Conservation Center in UP, and Fish Genome Conservation Center at UP, Lucknow. So while this can happen, while I am not denying that, the original strain, wild strains of India, either in the form of seeds or fertilized eggs or in any other form of life form, is being preserved, cryopreserved for posterity. Therefore, any point of time when you want a pure wild strain of a particular agriculturally important crop, we can get this from Delhi. There is another big organization called ICRISAT in Hyderabad. This is a World Bank assisted program. They also preserve wild varieties of several crops. So I think the concern is agreed, but I don't think that if, for instance, this kind of conservation technique man has had every time. You see paddy, paddy seed. Egyptians had the impression for the mummies, they fed their Maharaja with paddy. After 3,000 years, the seed has been taken to cultivate, the plant has come up. So if this is possible to conserve the seed of a paddy with the barrows, it should be possible for us with more scientific techniques. We have, I should not say many of these things, we have techniques of preserving seeds and spores, pollens, etc. Not only at minus 70 degrees, at minus 270 degrees. Also, government is now planning Ladakh, where it's always zero, to have a station where all these wild strain can be preserved, because you know electricity fails, so the whole thing program can go on. But God's grace, we have Ladakh, where we can preserve this forever. So all this likely to come. All my request for common man is, if this is something to do with transgenic, I think leave the problem to scientists. After all, we are also Indians. As a scientist, we look forward to preserve, conserve everything for India.
Sir, there is a opinion, at least some people believe that though in India we don't produce any transgenic crops, by importing food, we are eating transgenic food. For example, maybe coming as a part of a composition of a tinned food, or as a whole food itself. It's being legally, or legally we can't, but illegally maybe it's getting imported, and we are eating transgenic food. Is that true?
I do not know whether it is true or not, but we Indians are very good in planning. We are very bad in implementing. With reference to tinkering genome or manipulating genome, today what we are using as a poultry is we have tinkered with the genome, and it is so much used all over India, the poultry, chicken. I think the common man has a right to ask. The poultry people introduce steroid hormones to promote the growth of the chicken, and it reduces the cost of production.
Okay.
It is so also in milk production.
Okay.
The use of steroid hormones beyond a prescribed level is a legally punishable act.
Oh, yeah.
To my knowledge, either central government or state government have not yet made any legislation that in poultry farm you cannot use this steroids. You can use this particular steroid in particular dose.
Oh, yeah.
That kind of legislation has not come.
Yeah.
If you take America, we know that they have approved only 4 such steroids.
Okay.
And they have also approved the dose and duration of application. Duration is important because you can introduce these steroids during the early growing stage and leave the later half of it without any steroid. By the time the steroids are metabolized and excreted, so that when the chicken comes to market, it doesn't have any more steroids. This kind of scientific legislation has not yet come in India, to the best of my knowledge. But the common man, the civilians, have the right to tell the politicians, a government of India or government of any state, to bring in this kind of scientific legislation so that a common man need not suffer because of these steroid hormones. Here the common man has a right. I already said, whether it is transgenic plant or animal, or whether it is nuclear plant at Kudakulam, it should be left to scientists and engineers and environmental biologists. But at the same time, I like to say that the common man is a consumer of chicken or any other animal. If government closes its eyes to reduce the cost of production, if they are using enormous quantum of steroids, it will affect our new generation. It will tinker with the sex of the consumer, especially the children. So here common man has a concern, and it should be listened by scientists as well as parliamentary.
Today we have an eminent scientist and fish geneticist who was with us, and I thank you, sir, for your informative discussions. Thank you very much.
Thank you, sir. Thank you.